Sometimes, though, the environment could be completely outside the organism. Like that. Sometimes you can have, for example, a messenger from the outside world. What sort of messenger? A scary sight, a bit of sensory information, or whatever. Olfactory. Olfactory, suppose some odorant comes in, a pheromone. You're a female rat who has recently given birth, and the pheromones from your babies come floating in and bind to receptors here. Very similar principle again. And that causes them to activate something, and some cell down there. And eventually, there's a cell here that controls some muscles, and it makes your eyes dilate because you love the smell of your baby, and you're just, I don't know if rats have their eyes dilate, but humans will in many of those circumstances. Aha, how do you do that? You just change some structural proteins that did this or that, or suddenly this female is making some hormone like oxytocin as a result of smelling her baby. What have we got here? We got something going on in the outside world regulating what's going on with genes. Genes as the central dogma of life, as the information giver, nonsense. Events going on in the rest of the cell, the rest of the organism, the rest of the universe are determining when genes activate. So what we see now is a much more interesting world of regulation of gene expression. First, the modular ability for one gene to generate all sorts of different proteins, which brings up an issue after a while is, does that count as one gene? And people argue over that. And because of this 95% regulatory sequence business, the most interesting stuff going on with DNA is not what the protein is like, not what the protein does, not how well the protein does it, but when it does it, in what contexts. And what we've just introduced are if-then clauses into this whole world. If you get a signal that there is low glucose in the cell, then you begin to make proteins related to glucose uptake. If you have the smell of your child come in, then you activate this path. It's not changing what the protein is like, it's changing context. And I think what we will see is context is vastly more interesting than whether this protein is a little bit more like this, a little bit more like that. If it is being expressed instead at a different time, in a different place, in a different context, that's much more interesting.